have gave some basic information about the cell injury. What is the mechanism of the cell injury and what are the patterns of cell injury? What are the most two important types of irreversible cell injury? তাই তো এটুকু বলেছিলাম। So and I end up with the most important types of irreversible cell injury is necrosis. So I end up with the definition of necrosis. তাই তো what are the etiologies of cell injury? They are mainly of two types, one genetic causes, one acquired causes. What are the influences of the cell injury? What are the effects due to the adverse effect on the of any kind of influences over the cell, like cellular adaptation, reversible and irreversible cell injury and cell dying. The mechanism of cell injury, I have gave thorough detailed information about it, and the necrosis, the definition of necrosis. So I am starting my discussion today with the necrosis part. So first is definition. So what is necrosis? It is the death of a localized tissue followed by degradation of the dead cell due to the liberation of different autolytic, that is hydrolytic enzymes from the dead cells and dispersion of the dead tissue from the area. So basically this is associated with two things. One is liberation of autolytic, that is hydrolytic enzymes from the dead tissue itself, and second thing is inflammation. দুটো most important point কি হচ্ছে, along with the death of the group of cell within a tissue, there will be liberation of hydrolytic enzyme, first thing, and second is it is always association with the inflammation. That means different types of phagocytic cells coming in the picture, and they are phagocyting the or degrading the rest of the dead cells. তাই তো this is necrosis. So first হচ্ছে আমরা কি করে necrosis histopathology-তে চিনব। Whenever we do any kind of histopathology examination, we usually do the most important basic staining, which is known as hematoxylin eosin staining. Hematoxylin eosin staining. নাম শুনেছো স্টেনটার কেউ? নাম শুনেছো? So what are the two components of hematoxylin eosin? The name is already suggesting itself, self-explanatory name, hematoxylin and eosin. Eosin is very eosinophilic, so it is staining the cytoplasm, and hematoxylin is basic stain. It mainly staining the nuclear pattern. So two component of the cell, two component of the stain, hematoxylin plus eosin. Eosin staining the cytoplasm portion and hematoxylin staining the nuclear portion. So as if the nuclear portion is degrading due to the effect of different mechanisms which are involved in the irreversible cell injury. Here the irreversible injury is necrosis. So this nuclear portion is degrading. The amount of nuclear portion is decreasing. তাহলে hematoxylin বা basic stain, this is basic stain, this is basic stain, and this is acidic stain. Acidic stain eosin, whenever it stain anything, it looks pinkish, and whenever the hematoxylin stain anything, it looks bluish. So whenever there is decrease in the nuclear content, there will be decrease in the bluish tinge. bluish material within the cell will be decreased. আমরা কি দেখবো under microscope bluish material decrease করছে, and subsequently not like bluish, it's like purple color. So purple color, bluish color material decrease করছে, and there will be relative increase in the pinkish material within the cytoplasm. Relative increase in the pinkish material within the cytoplasm. That is known as increased eosinophilia. কেন হচ্ছে বুঝতে পারছো? So there is nuclear degradation. So nuclear content কমে যাচ্ছে within the cell. That means there will be relative increased amount of eosinophilic material within the cell, dead cell, and that is important term is known as increased eosinophilia. বুঝলে পয়েন্টটা? Second thing is the most important changes are occurring in irreversible cell injuries, nuclear. This is irreversible cell injury. Sign of irreversible cell injuries, there will be nuclear changes. তিনটে চেঞ্জ হয়, pyknosis, karyorrhexis and karyolysis. কোনটা আগে হয় কোনটা পরে, that doesn't matter. Actually all these three are occurring in simultaneous order. Simultaneously they are occurring. What do you mean by these three terms? First is karyolysis, means কিছু লিখতে হবে না, শুধু এখন তোমরা দেখো, আমি কিছু পয়েন্ট তারপর ক্লাস হয়ে যাক আমি বলবো যে একটা গ্রুপ করবে, সেখানে আমি কিছু মেটেরিয়াল দেবো, সেগুলোই তোমরা দেখবে। এখন জাস্ট শোনো, আমার কথাটা মন দিয়ে শোনো, কিচ্ছু লিখতে হবে না, যে যখন লিখতে বলবো তখন লিখবে, এখন কিছু লিখবে না। So karyolysis means there will be fragmentation of nuclei, fragmentation of chromatin. So the basophilia of chromatin is fades due to the fragmentation of chromatin, that is due to the breaking down of DNA. There will be the basophilia will be fading. কি বলেছিলাম, hemotoxylin strain কমে যাচ্ছে কারণ the nuclear content is degrading. It is known as karyolysis. Second point is pyknosis. Pyknosis means the whole nuclear content is condensed at a very small, significantly small portion. The whole content is getting shrinked. It is known as pyknosis. And karyorrhexis means it is whole nuclei is fragmented. So karyolysis means the fragmentation of DNA. Pyknosis means the nuclear shrinkage, and karyorrhexis means the nuclear fragmentation. These three changes are very important points of nuclear change, that is irreversibility of the cell injury. Okay. Third point key, of course there will be liberation of, as in the definition you have seen that there will be liberation of hydrolytic enzymes. কি করে লিবারেশন করবে, লিবারেট কি করে হবে, যখন সাইটোপ্লাজম, সেলের সাইটোপ্লাজম ডিগ্রেড করবে। দ্যাট মিনস শুধুমাত্র কি সাইটোপ্লাজমের মধ্যেই সমস্ত এনজাইমস গুলো থাকে তা নয়, এন্ডোপ্লাজমিক রেটিকুলামের মধ্যে থাকতে পারে, যে কোনো মাইটোকন্ড্রিয়ার মধ্যে থাকতে পারে, লাইসোজোমের মধ্যে থাকতে পারে, যে সমস্ত এনজাইম যেখানে যেখানে লোকালাইজ করবে সেখানে সেখানে থাকবে, এবং প্রত্যেকটা এনজাইমস অ্যাকচুয়ালি লিবারেট করছে। দ্যাট মিনস কি, all the cytoplasms, all the plasma membrane or the membrane of different organelles are degraded. So there will be breaking down of the plasma membrane and the membrane of other organelles. Third point. And due to this effect, there will be liberation of different activated hydrolytic enzyme. Activated hydrolytic enzyme, যেগুলো ডিগ্রেড করবে different organelles গুলোকে, as if গোটা সেলটাকেই ডিগ্রেড করবে। And of course this is always associated with the inflammation. যা যা অর্গানেল ডিগ্রেড হয়েছে, ভেঙে পড়ে আছে সেলের বাইরে, they will be phagocytosed by different inflammatory cells due to the initiation of the process called inflammation. Inflammation তোমাদের শুরু হয়নি, যখনই শুরু হবে তখনই বুঝতে পারবে যে whenever there is any kind of tissue death, cell death, the whole dead tissue or cell is scavenged by some group of cells. Those scavenger cells are known as phagocytosed cells, and the whole process is called inflammation. So necrosis is always associated with inflammation. And fourth point is, ওই যে প্লাজমা মেমব্রেন যেগুলো ডিগ্রেড হয়েছে, that plasma membrane forms some concentric laminated figures, concentric irregular laminated figures, as if they are look like this kind of figures under electron microscope, and they are called myelin figures. These are nothing but the remnants of different type of plasma membranes or membrane of different organelles. Okay, so these are morphological features of a particular necrotic cell. Now, what are the types of necrosis? কি types necrosis আসে, এইটা তোমাদের ধরেই নিতে পারো নেক্সট মানে সেমিস্টার, ইভেন নেক্সট প্রপের কোশ্চেন এটাই আসবে। What are the types of necrotic? What are the types of necrosis? There are mainly 6 types of necrosis. আমরা যখন আমাদের আন্ডার গ্রাজুয়েটে ছিলাম, তোমাদের বয়সে ছিলাম, তখন আমরা 5 টাইপস পড়তাম। কেন পড়তাম পরে বলছি। Now they are actually categorizing the necrosis into 6 types. One is coagulative, second is liquefactive, third is caseous, fourth is gangrenous, fifth is fat necrosis, and sixth is fibrinoid necrosis. Fibrinoid necrosis. একে একে আসি। ফার্স্ট ইজ কোয়াগুলেটিভ নেক্রোসিস। কোয়াগুলেটিভ নেক্রোসিস কি হচ্ছে এখানে? ইট ইজ দ্য মোস্ট কমন টাইপ অফ নেক্রোসিস। Almost in all solid organ, the term is very important, almost in all solid organs, commonly in heart, kidney and spleen, this kind of necrosis occurs. The most important etiology behind this necrosis is ischemia, hypoxia. So hypoxia, ischemia, prothome oxygen decrease, there will be decreased blood supply, and secondary to decreased blood supply, there will be decreased oxygen supply, so it causing the irreversible cell injury. Okay, now the death cell in this kind of deaths, in this kind of necrosis, type of necrosis, the death cell, the outline of the death cell remain as if the normal cell, the outline of the death cell is maintained. Suppose এখানে গ্রুপ অফ সেল ডেথ হয়েছে, এগুলো নর্মাল সেল ছিল, যার মধ্যে you can appreciate the nuclei, you can appreciate the normal organelles, cytoplasm, etc. Now this group of cells underwent death, and it is followed by liberation of different hydrolytic enzymes. So there will be damage to the plasma membrane, damage to the organelles, and of course there will be no nuclear membrane. So in case of coagulatic necrosis, the cell size will be raised, it will be raised, there will be bleeding formation, etc., swelling of the cells, the group of cells will be swelled up, and the outline will be fragmented. Why? Because there will be some pore formation within the cytoplasm, and there will be liberation of different hydrolytic enzymes, but there will be no nuclei within the cell. So group of cells look like this. A group of cell seen, they are all dead cell, and normal histology cannot be appreciated in this dead cell, but the architecture of the dead cell is maintained. This is known as tombstone appearance. Tombstone appearance. তোমরা জানো যে খ্রিস্টানদের যখন ক্রিমেশন হয়, তখন জাস্ট ক্রিমেশনের জাস্ট উপরে একটা টম্বস্টোন থাকে, একটা পাথরের স্টোন থাকে, যেখানে তার নাম গোত্র সব লেখা থাকে। So as if this is telling that something is there, some normal cells are there, but they are now not living cell, they are actually dead cells. So the architecture of the dead cell is maintained. Of course, there will be increased eosinophilia, anucleated cell, there will be no nuclei. So almost in all cases of solid organs, we can see this kind of coagulative necrosis. It's known as coagulative necrosis, except it is found in brain. সব সলিড অর্গানেই থাকবে, মোস্ট কমনলি ইন কেস অফ হার্ট, লাইক এক্সাম্পল হচ্ছে অ্যাকিউট মায়োকার্ডিয়াল ইনফ্রাকশন, নাম শুনেছো? কার্ডিয়াক অ্যারেস্টের যেটা কথা মানে সহজ কথায় বলে অ্যাকিউট মায়োকার্ডিয়াল ইনফ্রাকশন, ইটস অ্যান এক্সাম্পল অফ coagulative necrosis. There is a special type of area due to ischemia. When there will be some kind of ischemia, one part of area is in a solid organ is mostly affected. That area is known as infarct. See, this is an specimen of spleen. There is an wedge-shaped whitish area on the cortical portion. This wedge-shaped area is dead. কি করে বুঝতে পারছো? গ্রস স্পেসিমেন থেকে নরমাল লিভিং স্পেসিমেনে এরকম একটা রেডিশ ভায়াবল ভাইটাল টিঞ্জ থাকবে, রেডিশ টিঞ্জ থাকবে, which is not present. It is almost pale in color. This wedge-shaped paled area is caused due to death of these localized cells. Why it is occurring? Because there will be decreased blood supply, the end artery is obliterated and decreased blood supply to this wedge-shaped area. This is known as infarct. So in case of solid organ, when there will be obliteration of blood supply and portion of obliterated blood supply underwent some kind of coagulative necrosis, that portion is known as infarct. বুঝলে? প্রত্যেকে বুঝলে? Okay? So basically this is an coagulative necrosis. কোথায় কোথায় থাকবে? This infarct actually of two types. One is white color, one is red color or hemorrhagic infarct. White infarct কোথায় থাকবে? যেখানে solid organ থাকবে and যেখানে arterial blood occlusion হবে, artery occlusion হবে. রেড ইনফ্যারাক্ট কোথায় থাকবে? সেকেন্ড টাইপ অফ ইনফ্যারাক্ট, রেড ইনফ্যারাক্ট, এটা হচ্ছে হোয়াইট ইনফ্যারাক্ট। স্প্লিনিক ইনফ্যারাক্ট ইজ অ্যান হোয়াইট ইনফ্যারাক্ট। স্প্লিনিক আর্টারি যে এন্ড আর্টারি সেখানে অবলিটারেশন হয়েছে। ইট মাইট বি অ্যান অবলিটারেশন অফ মেইন আর্টারি অফ স্প্লিন, that is the spleen artery, splenic artery. But if there is any venous occlusion, if an area which is having dual blood supply, both arterial and venous blood supply, and both are obliterated, if the area is loose in texture like in brain, in that cases if there is some kind of ischemia, that ischemia causing the red or whitish infarct. এখানে যেমন দেখা যাচ্ছে কিডনিতে হচ্ছে এটা, ইটস অ্যান কর্টিক্যাল পোরশন অফ কিডনি। কিডনিতে আর্কুয়েট আর্টারি এর অ্যারেঞ্জমেন্টটা তোমাদের মনে আছে, অ্যান্ড তার সঙ্গে একটা সাইমালটেনিয়াসলি ভেনাসও মানে ব্লাড থাকে। সো দেয়ার উইল বি অবলিটারেশন অফ বোথ ভেনাস অ্যান্ড আর্টেরিয়াল ব্লাডস, অ্যান্ড দেয়ার উইল বি some reddish infarcted area seen in the cortical portion of the kidney. So this is all about infarction. Second type, now এটা আমি যেটা ছবিতে বলছিলাম, it's an schematic diagram, it's an photomicrogram under light microscope. Schematic diagram কি? এটা কি একটা infarction of the kidney. You can appreciate the glomerular structure, and these are the tubules. Suppose these tubular cells underwent necrosis. So see, there will be some kind of cells, and the cell, this dot nuclei are seen. This bluish nuclei now cannot be seen in dead tubules, but the outline of the tubules is seen, and relatively there will be increase in the pinkish tinge within the dead tubules. Okay, and there will be no nuclei. And in between the viable tissue and dead tissue, there is some kind of inflammatory cells. So this is an example of coagulative necrosis occurring due to the infraction of the kidney. Last bench, কি পড়ছো তুমি? আমি তো এখানে পড়াচ্ছি। What are you looking at right now? Next type of necrosis is liquefactive necrosis. The most important difference between the coagulative and liquefactive necrosis is there will be no boundary between the inflammation and dead cell. Why? Because the cellular outline is not maintained here. এখানে যে cellular outline গুলো coagulative necrosis এর ছিল, that cellular outline is not maintained here. So these dead cells and degraded organelles are gathered within a localized portion. কখন এটা হয়? When there is some kind of infective etiology. That could be viral infection, that could be fungal infection, that could be any bacterial infection. So whenever there is any microorganism, they are liberating extra amount of enzymes. বডির ডেথ সেলগুলো যা এনজাইম ছিল তার সঙ্গে সাপোজ কোনো মাইক্রোঅর্গানিজম যদি এক্সট্রা অ্যাক্টিভেটেড এনজাইমস লিবারেট করে, তখন কি করবে? এই যে সেলুলার আউটলাইনগুলো সেগুলোও নষ্ট হয়ে যাবে। The whole thing is gathered within a area which is full of viscous material, liquid viscous material, as if the death organ is present within a bag of liquid viscous material, and that is known as pus. এই যে তোমরা ছোটবেলা থেকে শুনে আসছো what is pus? পাসের মধ্যে কি থাকে? This degraded cells plus microorganism. তাহলে কি হচ্ছে? In this kind of necrosis, there is some kind of infective etiology. So there will be some kind of organisms within the dead area, and these organisms are liberating high amount of enzymes, and that enzymes are causing the lysis of all organelles, all kind of organelles, and as a result the whole tissue is converted into a some kind of viscous liquid material which is present within a bag, as if it is present within a bag. This is known as liquefactive necrosis. Liquefactive necrosis. It is mainly seen in the brain, central nervous system. It is mainly seen in central nervous system. See, this is an infraction in the cerebral cortex, and there is liquefactive necrosis. This area underwent liquefactive necrosis and it forms pus within the center. Surrounding the pus there will be so many acute inflammatory cells. Some kind of inflammation, acute inflammation is going along with that condition, and so many inflammatory cells are gathered within the area of pus formation. So this is liquefactive necrosis, the second most important type of necrosis. Third point is, এখানেও কি তাই দেখাচ্ছে? See, these are the cellular outline cannot be seen, and there is so many inflammatory cells and so many blood vessels are occurring. As a result of inflammation, there will be some amount of neovascularization. যখন ইনফ্লামেশন পড়বে তখন জানবে যে when there is any kind of inflammation, there will be some kind of healing also, and neovascularization might be there. So it is seen in case of necrosis in the brain. Third type is caseous necrosis. Caseous necrosis is specified form of coagulative necrosis. এখানে কি হচ্ছে? The cellular outline is maintained, but this is in periphery. At the center of necrosis, there is formation of cheese-like material. The name is suggesting cheese, caseous. The word comes from the cheese. So at the center of inflammation, there is some kind of cheese-like material, cheesy material, and surrounding the cheesy material, there will be aggregation of a specialized cell. These cells are tissue macrophages, which are differentiated into a specialized kind of cells known as epithelioid cells. Why it is called epithelioid cells? Because they are looking like the epithelial cells. They have slipper-shaped nuclei and they are elongated character with slipper-shaped nuclei. So surrounding this cheese-like material, there will be collection of epithelioid cells, and these epithelioid cells, in some cases, fuse with each other and forms a giant cell. কি হলো তাহলে? প্রথমে cheese-like material সেন্টারে থাকলো, তার চারপাশে collection of epithelioid cells, and some of the epithelioid cells fuse with each other and forms a giant cell known as multinucleated giant cells. It is very, very common in case of tuberculosis. In case of tuberculosis, the most important telltale histopathological feature of tuberculosis is formation of caseatin granuloma, and in case of caseatin granuloma, which kind of giant cell you can see? It is known as Langhans giant cells. These are formed due to the fusion of epithelioid cells. Multinuclear giant cells একই রকমের cellular structure থাকবে, slipper shaped nucleus, and those nuclei are arranged in horseshoe shaped pattern, as if suppose this is the cellular outline and the nucleus are present in periphery in horseshoe shaped pattern. It is known as Langhans giant cells. There are so many other types of multinucleated giant cells will be there. You will come across this kind of giant cell in the letter in your syllabus. Anyway, so altogether, and the whole thing is surrounded by collar of lymphocytes. And this whole structure is known as granuloma. And granuloma is found in caseating necrosis. কি হচ্ছে তাহলে? In this special type of necrosis, this is specialized form of the coagulative necrosis, there is formation of granuloma, which consists of a center of caseous material, which is surrounded by aggregation of epithelioid cells and some giant cells, and in some cases that might be surrounded by collar of lymphocytes. This is known as caseating granuloma, which is a feature of tuberculosis. Other than tuberculosis, you can find it in other so many cases, like in leprosy, like in syphilis, caseous disease. In some cases, this necrosis cannot be there, that is, this granuloma is present, but this caseation is not there. This is known as non-caseating granuloma. Granuloma হচ্ছে কিন্তু সেন্টারে caseation হচ্ছে না. It is known as non-caseating granuloma, like in Crohn's disease or sarcoidosis. Sarcoidosis এর স্পেশাল ফিচার কি থাকে? non-caseating এর সঙ্গে এই যে collar of lymphocyte, সেই collar of lymphocyte থাকবে না. This is known as naked granuloma. So naked granuloma found in sarcoidosis. It's a type of non-caseating granuloma, but in the surrounding the granuloma there will be no collar of lymphocyte. So this is a specialized form of necrosis known as caseating necrosis. Fourth type of necrosis is gangrenous necrosis. It's not a specialized form, but there is a typical type of cell death. There are mainly three types of gangrene. One is dry gangrene, one is wet gangrene, one is gas gangrene. See, this is an example of dry gangrene. This is usually found in the periphery of limbs. Frostbite দেখেছো, শুনেছো frostbite, অথবা অ্যাথেরোস্ক্লেরোসিস যাদের থাকে, হাই সিগারেট স্মোকিং যারা করে, অনেক সময় তাদের পেরিফেরিতে ডায়াবেটিক ফুট, পেরিফেরি লিমসের যে কোনো পায়ে বা হাতে পেরিফেরি আঙুলে কি হয়, there will be again obliteration of the small arteries. So the affected area actually went into this kind of form. They are looking bluish to actually blackish in color, and but the cellular outline is maintained. This is a specific type of gangrene known as this is a type of coagulative necrosis. It is known as dry gangrene. And treatment কি থাকে, এই এরিয়াগুলোর অ্যাম্পুটেশন করা, the viable tissue থেকে নন ভায়াবল টিস্যুকে সেপারেট করে বাদ দিয়ে দেওয়া। ওয়েট গ্যাংগ্রিন usually found in case of intestine. কি থাকে এখানে, হিউজ অ্যামাউন্ট অফ মাইক্রোব্যাকটেরিয়াম থাকে, হিউজ অ্যামাউন্ট অফ ভাস্কুলারিটি থাকে। সো a portion of segment of small intestine underwent type of necrosis. This is a kind of liquefactive necrosis. And last হচ্ছে gas gangrene. It is seen in case of infection due to Clostridium perfringens. Clostridium perfringens usually causing the necrosis of the muscle and subcutaneous tissue, and it forms some kind of gas under the subcutaneous tissue. See some blebs formation seen in the skin. So this is known as gas gangrene. Next type of necrosis known as fat necrosis. Fat necrosis is usually seen those areas where the fat content is high, like in case of breast, in case of pancreas, and there is liberation of enzyme known as lipase. Lipase কি করে, fatty acid and glycerol এ convert করে. That fatty acid actually binds with the extravasated calcium and forms a this is known as fat saponification. As a result of fat saponification, there is formation of some kind of chalky white or chalky salty material that is actually depositing within the necrosed fatty area. This is known as fat necrosis. Seen in case of traumatic breast abscess or acute pancreatitis, যেখানে the fatty tissues are affected mostly. And last type of necrosis is fibrinoid necrosis. এটা তোমরা আরো ভালো করে বুঝবে যখন ইমিউনিটি তোমাদের পড়ানো হবে। It is usually seen in type 3 hypersensitivity reaction, type 3 hypersensitivity reaction. কি হচ্ছে? It is mostly seen in case of the medial sized artery. Within the median sized artery, there will be deposition of immune complex, antigen antibody complexes. They are carried out by the bloodstream, and these immune complexes are deposited in the tunica media of median sized arteries, vessels of tunica media of medium sized arteries, and as a result of immune complex deposition, there will be secondary inflammation. So as a consequence, some kind of fibrinoid material, some kinds of leaky fibrinoid material liberated, and it forms this kind of appearance within the arteries. This is known as fibrinoid material. This looks like fibrin under the hematoxyl eosin stain, but this is not the actual fibrin. কোথায় পাওয়া যায়? Example is rheumatic heart disease known as Aschoff nodules, or malignant hypertension, or different collagen vascular disease like SLE. So কি টাইপ অফ নেক্রোসিস আমরা বললাম? কোয়াগুলেটিভ, লিকুইফ্যাক্টিভ, caseating, fat, gangrenous, fibrinoid. Now next point is apoptosis. This is the second type of irreversible cell injury. This is the main difference between the apoptosis and necrosis. কি হচ্ছে apoptosis এর মেইন পার্থক্য কি? Mostly the apoptosis is programmed cell death, as if the cell is committing suicide. Some kind of signal is there that particular cell has to be died, and that cell is underwent a specific kinds of chain reaction, and at the end of that kind of chain reaction, that particular cell will be degraded out. So this is an programmed cell death. This programming is actually maintained by nucleus itself. ভাবো সেল নিজে মারা যাচ্ছে এবং সেই গোটা প্রসিডিউরটা অর্গানাইজ করছে কে? নিউক্লিয়াস, and the whole cell is undergoing some kind of suicide. They are following a specific chain of reaction, and at the end of chain of reaction, the whole cell underwent degradation. Okay, so activated, there are liberation due to the signaling is from the nucleus, and due to the effect of that signal, some enzymes are activated. These are known as caseous. These activated enzymes which are causing the apoptosis, known as caseous, and that caseous causing the death of the cell. কি করে সেল ডেথ হয় তোমরা জানো? There will be nuclear degradation, there will be damage to the organelles and cytoplasms. But the most important thing is, these caseous are not damaging the outer plasma membrane, or in other words, I can say that there will be no liberation of hydrolytic enzymes. যদি হাইড্রোলাইটিক এনজাইম না লিবারেট করে তাহলে সেলুলার আউটলাইন কি থাকবে? মেইনটেইনড হবে না ডিগ্রেড হবে? মেইনটেইনড হবে। সো দেয়ার উইল বি মেইনটেনেন্স অফ সেলুলার আউটলাইনস, অ্যান্ড যদি সেলুলার আউটলাইন মেইনটেইন থাকে তাহলে ডিগ্রেডেড মেটেরিয়াল গুলো বাইরে বেরোচ্ছে না, দেন ইনফ্লামেশন হবে? ইনফ্লামেশন হবে না। সো দেয়ার উইল বি ফর্মেশন অফ সাম অ্যাপোপ্টোটিক বডিজ। ওই যে সেলটা ডেথ হলো, ডেথ হওয়ার পরে যে রেমনেন্ট পড়ে থাকবে সেলটাকে যে সেলুলার রেমনেন্ট পড়ে থাকবে তাকে বলা হয় apoptotic bodies. But in case of that apoptotic bodies, the cytoplasmic membrane will be intact and there will be no subsequent inflammation. So this is the most important features of apoptosis. See, there in case of necrosis, there will be, of course, there will be some kind of injury, but apoptosis is some kind of cellular cell death. The cell itself is committing suicide. Second point is there will be pathology. Some kind of pathology is there in case of necrosis, but in case of apoptosis, it might be pathological, might be physiological. There are a hell lot of example of pathological apoptosis, hell lot of example of physiological type of apoptosis. Third, in case of necrosis, we see some kind of cellular swelling, blebs formation, etc. But in case of apoptosis, there will be some shrinkage. The whole cell underwent in a very small shrinked bodies known as apoptotic bodies. পরে মরফোলজিতে আসছি। And of course, এখানে যখন নেক্রোসিসে আমাদের সেলুলার ডিগ্রেডেশন হয়, in case of apoptosis there will be no degradation. The whole chromatin material is condensed in a very small area. Mainly there will be pyknosis. There will be no karyolysis or karyorrhexis. There will be only pyknosis. Okay. And integrity of plasma membrane is maintained in case of apoptosis, and of course there will be no inflammatory reaction in case of apoptosis. So this is the basic difference from apoptosis and necrosis. কোথায় দেখা যায় apoptosis? Of course the group of the cell is damaged in case of necrosis, but apoptosis might occur in scattered cell, even in a single cell. What are the example? Physiological example key? Like in case of embryogenesis, involution of an organ, like periodic cycle of endometrium, shredding যখন হচ্ছে তখন apoptosis হয়। Next, hormonal dependent involution, like thymuses যখন involution হয়, that is an example of physiological apoptosis. Like self antigen, এটা আবার ইমিউনিটিতে যখন পড়বে, হোস্ট সেল অ্যান্ড সেলফ অ্যান্টিজেনের ক্ষেত্রে অ্যাপোটোসিস হয়। এখন বলছি না, ফিজিওলজিতে যখন ইমিউনিটিতে যখন পড়বে আরো ভালোভাবে বুঝতে পারবে। Next is pathological causes. Whenever there is a particular cell is injured, there might be, mostly they are viral infected cells, there might be an apoptosis or some kind of pathological atrophy. Suppose there will be decrease in the vitamin, decrease in the small nutrients, micronutrients within a body. Senile atrophy of endometrium, senile atrophy of brain, these are example of pathological apoptosis. Okay, then tumor cells যখন কোনো নিউপ্লাস্টিক সেলস যখন underwent, mostly they underwent kind of pathological apoptosis, or it might also occur along with an necrosis known as coexisting apoptosis along with necrosis. So apoptosis এ mainly 3 phase থাকে, 3 phase. First phase is known as initiation phase, second phase is known as execution phase, and third phase is known as degradation phase. Initiation phase is tightly regulated by, কি বলেছিলাম, DNA, that is nucleus, the nuclear content, and they are actually organized by activated enzymes known as caseous. Second হচ্ছে execution phase. Whenever there is some kind of initiation, যখনই কোনো একটা প্রসেস ইনিশিয়েশন হয় তখন তাকে শেষ করতে হয়। কি হবে, প্রসেসটা একটা সেল ডেথ ডাইং ফেজ শুরু হয়েছে, সেটাকে শেষ করতে হবে, তাকে বলা হয় সেল ডেথ। That activated caseous bring the cell death, and that dead cells should be removed from a site of origin, that is known as degradation. Degradation or dead cell scavenged is done mainly by phagocytosis, process of phagocytosis by some tissue macrophages. The tissue macrophages are known as phagocytes. So first phase is initiation phase. Initiation phase can be of two types. One is extrinsic pathway, one is intrinsic pathway. Extrinsic pathway, in case of initiation phase, extrinsic pathway is mainly caused by the 8 or 9 and caspase 8 or 9, and execution is mainly caused by 3 and 6. So mainly of two type initiation, one is extrinsic pathway, one is intrinsic pathway, and there are two type caspases are seen. These are metabolically active enzymes. Initiation is mainly done by caspase 8 and 10, and execution mainly done by caspase 3 and 6. Besides these activated enzymes, there are some important genes and genetic products, mainly proteins. So whenever there is mainly in a living cell, these two type of genes and genetic products maintain an balance. A normal homeostasis maintained between these two genetic products. These are known as pro-apoptotic factor, anti-apoptotic factor. সোজা বাংলায় বলতে গেলে, if that pro-apoptotic factors, the level of pro-apoptotic factors increase, বা যদি সেলের মধ্যে pro-apoptotic factor বেড়ে যায়, then the cell will die, underwent apoptosis. যদি anti-apoptotic factor বেশি থাকে, যেটা নরমালি থাকে, তাহলে cell will prevent the process of apoptosis, prevent the process of initiation phase. So what are the example of pro-apoptotic factors? These are mainly genes. Again I am telling these are mainly genes, and when I am telling the factor, then it is denoting the genetic products, that is its protein. So what are the examples of pro-apoptotic factors? Vax, BAC, BIN, BID, 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 PUMA, P53. P53 is known as guardian of genome. It's a very important genetic gene. পরে তোমরা জানবে আরো, again এটা যখন নিউপ্লাসিয়া হবে তখন আরো জানবে. These are all pro-apoptotic factors. They actually initiates they giving the signals that the cell should initiate the initiation process of apoptosis. And what are the anti-apoptotic factors? BCL2, BCLXL, MCL1. So initiation ki kore hocche? Extrinsic pathway. Extrinsic pathway, there are two types of initiation. One is extrinsic pathway. Extrinsic pathway is mainly known as fast ligand mediated pathway. This fast is present in the outer cell membrane or outer cytoplasmic membrane. It might be present in the mitochondria, it might be present even in the cytoplasm, okay, in any kind of organ in the outer membrane, outer bilamellar membrane, outer membrane fast thake. So whenever there is some kind of apoptotic signals, this fast cross-linked, they cross-linked with each other and they binds with some ligand known as fast ligand. Three fasts actually cross-linked with each other and bind with the fast ligand. Sei fast ligand ta ki? It is known as fast associated death domain. Fast associated death domain. This is present in the inner membrane, like inner mitochondrial membrane. So ligand binds with the fast. Ligand ta ki chilo? Apoptotic signals. Sei apoptotic signals, fast ligand, known as fast ligand, they bind with the fast. There will be polymerization of the fast, and fast from outer membrane binds with the fast associated domain, FAD present in the inner membrane. মানে ব্যাপারটা হচ্ছে এরকম, এখানে অ্যাপোপ্টোটিক সিগন্যাল আসলো, they liberate some kind of ligands bind with the fast. Due to the binding, there will be polymerization of 3 monomer of fast. This is present in the outer membrane. They bind in the fast ligand in the inner membrane. It is known as FAD. So this is fast polymerization, this is fast ligand, fast polymerization, and they bind with the FAD, and this activated FAD actually activates the caseous. In case of extrinsic pathway, the caseous 8 or 10 is activated. So this activated caseous in the downstream activated so many other caseous. এটা মনে রাখতে হবে না যে what are the other caseous actually activated in the downstream. Only remember there will be multiple pro-caseous cleaved each other. It's a chain reaction. Again I am telling, activated caseous 8 or 10 in the downstream maintain a chain reaction, and so many other pro-caseous are actually cleaved and form the activated caseous, and active caseous, execution caseous are formed. So this is one type of extrinsic pathway. This another type is intrinsic pathway. Again, in case of intrinsic pathway, there are maintenance of these two factors, that is anti-apoptotic and pro-apoptotic factors, is that balance is lost. There will be, of course, pro-apoptotic factors, the level of pro-apoptotic factors within the cell is increased, the level of pro-apoptotic factors increased, and the level of anti-apoptotic factors, of course, there will be decrease in the anti-apoptotic factor. So if this condition occurs, যদি এই imbalance দেখা যায়, that cytochrome C which is present in the inner mitochondrial membrane, they will be leaked out, and that leaked cytochrome C, they binds with the apoptotic activating factor, or known as APAF1. Apoptotic activating factor present in the cytosol. So cytochrome C from the inner mitochondrial membrane liberate into the cytosol, and within the cytosol they binds with the APF1. এখানে যেমন ফার্স্ট লাইগ্যান্ড ফ্যাডের সঙ্গে বাইন্ড করে ক্যাসপেস 8 আর 10 কে অ্যাক্টিভেট করছে, এখানেও অ্যাকচুয়ালি একই রকম ভাবে cytochrome C, it's leaked out into the cytoplasm and they bind with the APF1 and they actually activate the caspase 9. Okay, caspase 9. So it causes the execution. So ultimately they are again same J in the downstream they processed so many caspases into active caspase, and those active caspase actually causing the execution. Ki execution korbe je they are mainly mediated by the activated caspase 3 and 6. Ki execution korbe they actually provides the different kind of hydrolytic enzymes. These hydrolytic enzymes cause the damage to the cytoskeleton. These hydrolytic enzymes cause the damage to the nucleus chromatin. So different organelles and the nuclei are degraded. They become non-functional. They become non-functional. The normal cytoskeleton is lost. The normal nuclear content has been lost. But of course, the outer plasma membrane maintained. সেটা কিন্তু ড্যামেজ হচ্ছে না। ওকে, and তাহলে কি হবে? The whole cell is converted into a small shrinked cell. Within the shrinked cell, the chromatin is condensed to a very small portion known as pyknosis, and the cell which underwent the apoptosis known as apoptotic bodies. Last phase ki hobe? Degradation phase. This is actually maintained by degradation of that apoptotic bodies is done by the process of phagocytosis, and this process of phagocytosis is actually organized by the macrophages. These are tissue macrophages. Okay, so কখন চিনবো টিস্যু ম্যাক্রোফাজ, কখন চিনবে যেটা নর্মাল সেল নয়, এটা অ্যাপোপ্টোটিক সেল? কারণ টিস্যু ম্যাক্রোফাজ তো নর্মালি সমস্ত জায়গায় থাকবে, তাহলে কেন একটা অ্যাপোপ্টোটিক বডিজকেই শুধুমাত্র টিস্যু ম্যাক্রোফাজ চিনতে পারছে? কেন কি there is something known as phosphatidylserine. Phosphatidylserine is present in the inner mitochondrial membrane, but in case of apoptotic body, this phosphatidylserine is going into the outer mitochondrial membrane. This is known as serine flip. This is known as serine flip, and this antibody to the phosphatidylserine is known as CD95 or annexin. This is an antibody to the phosphatidylserine known as annexin 5 or CD95. This is the marker of apoptotic cells, and whenever there will be phosphatidylserine in the outer cytoplasmic membrane, they will be scavenged by the phagocytose. They actually acts as an ligand for the phagocyte tissue macrophages. So tissue macrophages binds with that phosphatidylserine, and this is a marker of apoptotic bodies. So of course there will be no inflammation and no kind of other inflammatory cell at the site of injury. So last, how can you identify this is the group of cell or a scattered cell is undergoing some kind of apoptosis? Ki hobe? Apoptotic cells are very small, round cell. They are very shrinked, very small, and the nuclear content is condensed within a very small portion. There might be some kind of DNA breakage, but it's a very rapid process, and it is very difficult to see under the light microscope. কি বলেছিলাম তাহলে আমরা চিনতে পারবো কি করে? How can I identify this might be an apoptosis cell? Because there will be some marker known as annexin 5. So in electron microscope there will be some kind of chromatin condensation, shell shrinkage, cytoplasmic belts, and apoptosis body formation. And in case of DNA chromatography, they are in case of polyacramide gel electrochromatography. In case of PAGE, the DNA forms in a, that is not the DNA, this is the degraded portion of the DNA, and they form a ladder pattern in case of page. Ladder pattern in case of page, this DNA breakdown is visible as a DNA ladder in case of page. And due to the activation of these caspases, mainly it causing the damage to the DNA, so some kind of activated DNAs can be found in case of blood.